Okay, I make that the hour, so I think we'll begin. Welcome everyone. Good morning, good afternoon, good evening, wherever you are. It's great to have people from all over the world today. My name's Chris Thorne. I'm the Senior Manager of Field Marketing here at Twist Bioscience, and I'm going to be hosting today. So before we begin, just a little bit of housekeeping. As ever, you'll find yourselves all muted. And this will be the case for the whole of the webinar. But if you do have questions, please do use the Q&A box at the bottom of the screen rather than the chat box, and we'll be taking some time at the end for a Q&A. Following the webinar, we're going to send you to a short survey. So if you do have time, it'll take about a minute, and this just helps us understand how we've done on this particular webinar and whether we've delivered on what you were hoping for. So please do take a moment to complete that. Okay, so it's my pleasure to introduce Marcel Nelen. Dr. Nelen is Deputy Head of the Genome Diagnostics Division at Radboud UMC, a clinical laboratory geneticist, and also heads the Radboud Genome Technology Centre, a facility unit specialised in DNA sequencing using automated and innovative high-throughput laboratory technologies and workflows. Genome Diagnostics at Radboud UMC was the first diagnostic laboratory to apply exome sequencing in a clinical setting. And since then, the core facility developed several innovative clinical workflows using various sequencing platforms. So Marcel joins us today to talk about their automation of mechanical fragmentation and the Twist exome workflow on Hamilton. So Marcel, I'm going to hand over to you.
Okay, as Chris already explained, we're going to share some information about clinical rapid exome sequencing and the workflow that we developed at the Radboud University Medical Centre for using twist and enrichment, and to see how things go. I will give you some insights on how we organized ourselves and how we organized the logistics, and I'll take you through some of the steps during the validation and some of the background information that is probably needed if you want to repeat the workflow in your own laboratory. First, some background information. This is a slide that shows a picture of an artist representation of the first female that was sequenced. That's a statue that is on our campus. This is a representation of the genome of one of the clinical geneticists here in the Netherlands that was sequenced as the first female. But it's nice to have such a statue on campus. Human genetics department is quite a big department. It's about 350 people in total, covering both diagnostics laboratories, research laboratories, and of course clinicians who see all the patients. The Radboud University core facility for next-gen sequencing is embedded in the genetics department, and roughly 22 people are working in this core facility, both staff and technicians. We are quite clinically focused, so roughly 80% of what we do has a diagnostic purpose in all the testing and the sequencing that we do, and more or less 20%, so the overcapacity of the machines that we have is used to also support research sequencing and other departments within the University Medical Centre. Probably 10-20% of what we do is coming from abroad, so that also helps to make some money to sustain all the machines and all the equipment that we need to buy to update ourselves to the most to the best new machines that are available. Roughly 25,000 DNA isolations annually and 16,000 reports are produced within our department. And we have a very high degree of automation. So I think we already have 19 Hamilton robots available that support all the workflows that we developed over the years. We have a strong focus on generic workflows. So we we like to have workflows up and running that sustain multiple technologies and indications within the department. And with that, we also started a few years ago to strongly collaborate with Maastricht University Medical Centre. In fact, we have the same head of the department at both locations, so that's quite unique, I would say. Here you see a representation of, or at least this is probably the beating heart of our facility. This is all the platforms that we currently have up and running in our facility. Here in the back, I don't know if you can see my pointer, but here in the back you see good old-fashioned ABI machines. Oh, sorry. And here you see 2 NovaSeqs, which nicely contrast with the good old-fashioned ABI machines for Sanger sequencing. This is the more modern machine. So the rapid exome sequencing is run on the NovaSeq equipment. You see also 4 PacBio machines on the right side of the picture. And you see on the left side some NextSeq 500 machines that we currently run mainly for research purposes. And if you look carefully, you can also see a BioNano Sapphire machine in the picture. So this is essentially the beating heart of our facility, and these machines sustain all the workflows that we have of support. As Chris already mentioned, in 2011 we started with clinical exome sequencing. That was late 2011. Actually, in 2012 we did the first 500, and then eventually that grew to about 6,000 exomes annually, and that is more or less the number that we've seen over the years within our hospital. And in 2016, we decided to also start with a workflow that would help neonatal care units to get rapid information for patients that were present at the NICU sites. That was introduced in 2016, and then over the years this quickly increased, and it became apparent that there are a lot more patients in need for a rapid outcome of the tests, and we said we will use the rapid exome workflow essentially for all patients that are in a crisis situation. And now we're running roughly 60 samples on a weekly basis, becoming quite a serious workflow in our facility at the moment. So automation is needed when you start running these numbers, but the initial runs in 2016, 17, and 18 were mainly done on a manual basis. A quick explanation of how we perform clinical exome sequencing. In our laboratory, it's a 2-step process where for all indications, exome sequencing is essentially the basis for the analysis. But we create virtual gene panels which we use to analyze the data. So the first step is a gene panel analysis depending on the indication or the phenotype of the patient. It can be blindness, it can be deafness, it can be muscle panels, or intellectual disability, which is a very large panel. If that is positive, of course, we write a report on this positive outcome. If it's negative, then it depends on the consent that is given by the parents or the patient. if we proceed to look at the entire information that is present in the exome, and then we look outside the panel to all genes, and then we probably identify new genes or new phenotypes that are not thought of, but essentially that helps getting the diagnosis of the patient in quite a substantial number of cases. And with rapid whole exome sequencing, essentially what we're doing is more or less the same strategy, but now in a high pressure cooker. So what we do and normally do for the bulk of our exomes, we have 30 to 60 days and sometimes even longer to analyze, to run the sequence, but also to analyze the data, and we have quite some time for that. But if you run a rapid WES, you want to produce a report, let's say from sample in to report out within, let's say, less than 10 days. So routinely we have 7 days, but every now and then it takes a bit longer, and then we have at least 2 weeks or so for having a report written. So that is essentially what we do. We just put a lot of the effort in speeding up the sequencing and speeding up the interpretation of the whole process. And if you want to do that, then logistics is key. So initially, when we started with the rapid WES procedures, we expected not too many samples, so maybe a few a month. And then we said, okay, just give us a call, and if we know when the sample arrives, we start working on it. And so we had to discuss and plan quite a lot before we could actually run a sample, and to make it cost efficient, we usually waited until we had multiple samples. That produced quite some differences in our turnaround time, and we wanted to have that more predictive. So let's say the middle of 2017, we had a discussion with the team, and then we decided to have a fixed starting point. So we would run, regardless of how many samples we had, on a weekly basis, a run for our rapid exomes. And we just told the clinicians and the people involved in requesting these rapid WES that we would like to have a sample on the Friday before noon, and then we had the afternoon still available for DNA isolation, and then we would start working on it on Monday and doing the library prep, and then start the sequencer on Tuesday. Usually the sequencing would be finished on Wednesday, and then Wednesday overnight the bioinformatic pipeline would kick in, and then on Thursday and Friday the data would be available for interpretation. That worked until recently very well, even with our manual workflows, and that eventually produced quite a constant turnaround time of roughly about 10 days, even with the increase of the number of samples. And while we were doing that, there was quite some discussion in the lab if we would start running twice a week, because that would even give us a more easy workflow, and also we would probably have less discussions on if the sample would arrive on time and things like that, because if the sample comes in, it doesn't really matter. The maximum waiting time would then be 2 or 3 days or so before the library prep would start. So there you have a more constant logistic process that would help the workflow. So this is the workflow that we've been running for the last 2 years or so, 3 years, and that helps tremendously to get a more or less constant turnaround time. But with the increase of the volume of samples, we needed to think about automation of the workflow, and that is also coinciding with looking at new enrichment strategies that we had. So we had a discussion with several companies, also providing new equipment and new technologies that became available. With our rapid exome, we were using enzymatic shearing, and in the facility we actually noticed that the enzymatic shearing was more vulnerable for failure. We receive a lot of DNA samples from external parties, and they use all different DNA isolation methods with different buffers, and that was giving us a reasonable number of failures in our exome workflow. So we decided to start using equipment from Covaris, which developed a new mechanical shearing device called the R230, and that is an on-deck machine that you can put next to your robot or in your robot, and that could actually help us performing the mechanical shearing, and we feel that is a more constant procedure, and we have less failure in our workflow. We also have an on-deck thermocycler, of course, and multiple pipetting heads can be used to support the workflow. But the upper picture essentially shows the setup that we use. The entire workflow is traced by our LIMS system that we use, LabVantage. And so we use barcodes and everything to keep our errors to a minimum. The capacity of the workflow is now 32 samples per run. We can expand to about 64 samples per run, but then the working day of the technician becomes a bit longer. So we probably have to change our workflow a little bit, and then we could run, let's say, twice 64 samples per run. And then on a weekly basis, we could run 4 runs, because we now have, since 2 or 3 weeks, we have now a second robot system up and running. So we have quite some capacity now to increase the rapid exome workflow. Our initial test with the twist exomes already originated in 2017, late 2017 and early 2018. We were testing a twist, and at that time they only had the core exome available. And I wrote down the main conclusion of our bioinformatic team and the analysis that they performed. And they told us that the twist enrichment kit actually looks good in terms of mapping reads and evenness of coverage. Even 20x coverage ratio per target was really well. However, with the core exomes, we were actually missing some crucial coding regions, which would create potential issues when if we wanted to use this in a clinical setting. So at that time, we decided not to go ahead with twist and just wait a little bit to see if they would improve their core exome. And eventually in 2018, a new kit became available, was an extension of the core exome using RefSeq sequences. And that made a substantial difference in how the exome performed. Here you see some of the standard QC that we checked, again with the manual evaluation of the twist exome. we've sequenced actually quite deep to a mean coverage of almost 300x, which is of course bizarre, but we were testing it with just a few samples on the flow cells. So that also explains probably why you see these crazy numbers on 30x coverage. But overall, the data was really well, and we were really pleased to see that also the number of exonic single nucleotide variants increased with almost 3,000 SNPs. So that gave us enough confidence that we should continue to test the twist exomes. Here you see a slide that shows more or less why we were pleased to use twist. In the lower part of the cartoon, you see our standard workflow, and you can clearly see, if you compare the 2018 run with the 2019 run, that some of the exons were missing in our standard workflow for this SPEST gene. So exon 13 and 14 were actually missing. And with the twist exomes, both with the core and with the RefSec addition, you can clearly see that these exons are now very well represented in the data. Here's another example in a gene called SEP290, where you again can clearly see that some of the exons were missing in our standard workflow, and you can clearly see that they are represented in the data that we produced manually in 2018 and on the robotic systems in 2019. So is it all hooray, hooray? Not all. We also see some difficulties, especially with switching to a robotic workflow. Here you see the two runs that we performed on the robotic system, and you can clearly see in the middle part of the cartoon, so with run 5, that exon 1 of the silinon gene is actually missing in the data, which was present in the manual workflow that we tested in 2018. Differences are, of course, robotics is used, and enzymatic shearing in 2018 compared to mechanical shearing. Tom, of course, did his magic using the robotics and all the knowledge he had, and in discussions with the team, we eventually tweaked the workflow to such an extent that we could see again data of exon 1, although it's still not as well represented in 2018. But overall, the exomes perform really well, but there are still some regions that we need to improve on. So this gave us enough trust to start validating our workflow and start validating the robotic workflow. So we gave some special attention to two quality criteria that we think are really important, especially working with new enrichment strategies. One was the evenness of the data in our initial testing. So the core the core exome from twist performed really well in terms of evenness. Adding the RefSec genes to the core enzyme actually showed that we lose a little bit of the quality in terms of evenness, but it was still performing better than some of the competitors. And the other thing that we noticed was that the on-off target ratio of map read was actually less than we would normally see. So in our standard workflow, we noticed routinely almost 90% on-off target, and with twist exomes, we clearly see that we're lower than let's say 75%. So that needed some attention, and we looked at especially at these two criteria with let's say extensive interest. So in terms of evenness, we used the knowledge of Twist as a company, and particularly the pipelines they have running in their systems. So we extensively worked together with Tina, and Tom basically had a hotline every time when new data was produced, and the data was analyzed, and we were particularly interested in the Fold 80 score. And that's a score that we were actually using to see if the evenness of the data, so the uniformity of the data is really is good, and what we would like to see is a Fold 80 score of around 1.4, and the Fold 80 score actually is a measurement of the mean depth divided by the 20th percentile. Giving you more or less the explanation that if you would need 1 million reads to reach 30x, Fold 80 score of 2 would mean that you would need 2 million reads, and a Fold 80 score of 1.4 would give you more or less the same data set if you would use 1.4 million reads. So it tells you a lot of about the evenness, and if you're actually over sequencing with your exomes. So this was something that we checked quite regularly, and we aimed at a Fold 80 score of about 1.3, 1.4. That looked really well. And we manage that now on a routine basis. The other thing that we really looked at was our on-off target percentages. We noticed, as I already mentioned, that Twist was performing less compared to our standard workflow. And here you see a cartoon of that. And we thought about this quite heavily, and we tested quite extensively at trying to tweak the robots to such an extent that we would reach 80% or so. But that turned out to be fairly difficult, and we think we have identified the reason for that. Actually, what we noticed when we start looking around in some of the exomes that we produced, we noticed that in other areas than we were expecting data, we see quite some regions that actually have a fairly high coverage of reads. As you can see in the upper and the lower part of the cartoon on the right, it shows the two Twist versions, so the core and the RefSec added version. And there you can see clearly sequencing reads on regions where no targets are located. And we asked Jordi, our bioinformatician, if he could look for regions with over 300 reads without any targets present, and he identified at least 1,300 reads that shows this type of sequencing data. We asked around if other people experience more or less the same thing, and we also looked at data from competitors, and also other enrichment strategies actually have the same show the same feature. So it's not something that is unique for the Twist exomes. We've looked at Roche data that is showing more or less the same data. We looked at Agilent data that gives more or less the same data, especially in the kits that have these large RefSec data set included. So it's not unique, but explains why you find a much lower on off-target value than you would normally expect. Some numbers. Here you see the validation. We looked at the single nucleotide variants, and we looked at how the twist RESIC exome compares to our standard workflow. We would expect more or less 120, 130,000 single nucleotide variants, and we see that over 105,000 of these variants are actually overlapping, so identified by both enrichment strategies, so the standard workflow and the twist workflow. And what we see is that there's quite a substantial number of single nucleotide variants that are actually unique for both enrichment strategies. So the roughly 30,000, 31,000 variants are unique for the standard workflow, and 26,000 variants are unique for the twist exome, which is not strange. You expect that, of course, but what is more important, are these actually interesting enough, and are these true variants? So we looked at the GATK score, which is an indication of how well these variants are true. And from the 31,000 variants or so in our standard workflow, 12,000 are actually true variants, as we would call them. So with having a score of over 500, so which is normally a score that we would use to not confirm variants with another technology like Sanger sequencing or so. We looked at the same data set for the twist exomes, and also there we see over almost 10,000 variants that are actually having a very good GATK score. But what we were really pleased with is that we saw that over 3,000 or almost 3,000 variants were actually exonic in the twist exome, and only 400 or so were exonic in the routine exome that we produce. So we did clearly see an increase in the number of variants that represent exonic sequences. So clearly we have now a more complete and probably a better exome than we used to have with our standard workflow. Both of the strategies actually produce a very nice bimodular distribution of the variants. So you expect a lot of variants to be homozygous, and you expect a lot of variants to be heterozygous, and that is clearly visible. We also looked at CMV detection. So we are routinely looking at CMVs in our exome. We don't have that many QC criteria that we really can look at, but one of the important ones is the number of variants and compare that to our autosomal standard deviation. And what we would expect is that the CMV, the calling of the CMV actually with the standard deviation are in between the two horizontal bars, and very near the vertical red bar that you see in the cartoon. We clearly see that the twist exomes perform well. They are probably a little high in terms of standard deviation, but it's clearly better than the standard workflow that we have. So the evenness probably helps quite a lot in calling these variants. So looking at all these criteria that we set at the beginning of the validation, we can now safely say that they basically ticked all the boxes, and there's no reason not to start using the twist in the clinical workflow. So from May this year, we started to use the twist RefSeq added exome in our clinical workflow, and we've collected already, let's say, I think we've run over 800 samples now, and this is a representation of the first 350 or so exomes that we run in the first few months. We noticed a few things. The top left cartoon actually shows the mean coverage. You see quite a variation, so we need to improve in how we make the pools, and we probably have to tweak that a little bit so we get a more constant and more efficient running of the filling of the flow cells. But overall, the samples rarely fail. So the aim is to have at least 80x, and that is what we quite routinely see. We have a few exomes that are lower. On the right, the top right, you see duplicate reach, which is probably one of the more important QC criteria for the complexity of your exome, and over time you see a little bit of an increase in the duplication ratio, but we tweaked the robot a little bit when we noticed this trend, and now the samples are more or less back to normal in terms of percentage duplicate reads. So we expect 6 to 8% duplicate reads, which is really nice. On the left lower part, we see a QC that we think is actually more important than we originally thought. So the percentage 20x coverage is actually really constant if you look at the depiction of these first 350 exomes. But it tells you a lot. So this tells you a little bit on the evenness of your data. And recently we identified 2 samples who actually had a percentage lower than 90%, and actually these failed. But all other QC values that we check were actually normal or fulfilling our criteria. So this was the only QC that we followed that actually failed and gave us an indication that. So this is a QC that we just recently added to our list of important QCs. So my, let's say, recommendation would be that everything above 95% would be acceptable. Let's say, and everything between 90 and 95 probably would need a warning. Check your data. Everything below 90% would be a failure. And then on the right of the cartoon, you see something that Tom recently saw and observed in the data. You see a representation of the insert size compared to the on-off target that we measure. And we, like I said, we normally see anything between 70 and 75%, but every now and then we see samples that do match, let's say 80% or even more. And apparently these coincide with samples that have a somewhat shorter fragment size. So that is something we observed recently. And we think, well, we need to see and discuss this if this is really important. What we do know is that the majority of the samples that have these lower insert size, that these are prenatal cases. So it might be important, but we need to follow up on that. So what this transition to twist exomes brings us, well, one of the conclusions is probably obvious. It gives us a better quality and a more complete exomes than the standard workflow that we had running. Automating it allows us increase in sample size and volumes quite easily without too much effort in terms of FTE that is needed. And it allows us to start running twice a week, which helps to comfortably say that the turnaround time will be probably close to 10 days, with the exception if the run really fails with bad library production. But 10 days would be fairly constant. We see an increase in indications that are making use of the rapid WES workflow. For instance, Noonan syndrome was a workflow that we already had up and running in our exomes, but we still needed to sequence, I think, 9 or so Sanger amplicons to make it a complete analysis. Now with the twist exome, it's actually finished in one run, and we don't have to Sanger or add Sanger amplicon based sequencing to get a complete analysis of all the genes involved. We see quite an increase in the number of prenatal cases. Both in let's say just routine exome sequencing, but also for CNV analysis. This becomes more and more the idea to start using the rapid exomes, and other groups are thinking about adding new indications to this workflow. So we expect that the sample size will increase quite substantially in the next months or so. But also not unimportant, it reduces the sequencing costs. In our standard workflow that we used to run, we would run 16 to 32 samples. Depending on the flow cell, so the SP flow cell 16 and 32 as one flow cell. Using twist actually allows us to reduce sequencing costs by half, because we can double the amount of samples per flow cell. So on an SP flow cell, we routinely run 32 samples, and on an S1 flow cell, 64 samples, which helps tremendously in saving costs. But what we hope that it definitely would bring is that we will shorten the diagnostic odyssey of the patients, particularly in crisis situation. So neonatal care, postnatal cases that need special attention, but also prenatal cases where we see all kinds of fetal abnormalities. We have analyzed the data set last year or so when comparing all the sequencing that we've done, and roughly 20 to 30% of the cases we will identify a causative or a likely causative mutation in the data. And if you would add possibly or variants of unknown significance to that, but with a likely suspicion of being pathogenic, then numbers even increase. So opening up this rapid exome workflow to a larger group of patients would definitely help to speed up the diagnostic process and to get hopefully a positive outcome in terms of a diagnosis. With that, I'd like to thank, of course, everybody involved in this workflow, particularly Tom Hofstee, who is for sure a driving force for setting up this workflow. He is incredibly knowledgeable for using Hamilton robots, and we're really pleased with the results. Of course, also the bioinformatic team, particularly Jordi Kourouminas Galvani, who had a direct line with Tom when all the data needed to be analyzed. Incredibly important to get a well-validated workflow. And Sanne Smekens, I think, needs also a very big thank you, because she mined all the variants for relevant and interpretation of all these variants to see if the workflow would really meet the diagnostic criteria, and everybody else involved. So also Covaris and Hamilton, of course, for all the support that we got and enabling us to do beta testing of the sharing, the R230 machine, and Twist, of course, particularly Brian, for helping us setting up all the meetings and discussing what we could do, and Tina Hahn for helping Tom with all the analysis of the exome data. And with that, I'll be happy to take all questions that you have. Thank you very much.
Thank you, Marcel. That was fantastic. Really appreciate the talk. And we do have a number of questions coming in, so I will start working my way through those now. I'm just going to bring them up to my other screen. So predominantly quite technical questions, so I hope you'll forgive diving back into the data. So for example, on one of your slides, you showed that the change in the duplication rate. A question here from an anonymous attendee asking if you had, or what part of the automated workflow did you have to modify to counteract the increase of the duplication rate? Was there something that changed there?
That's a question that Tom probably is able to answer more than I can do, but likely this has to do with some of the washing and the bead purification that is needed during the enrichment. And there we have tweaked quite substantially in the workflow to get that organized well, and also timing that the pipetting steps take is actually quite important.
Okay, so just, I mean, for my benefit then, just so the workflow that you have now is still constantly undergoing optimization and improvements, or is it set it now, or?
Well, it's more or less running a constant, but we do perform trend analysis, and if we see things change, like the duplicate reads that we saw over time, then of course we check the things, and we check all the consumables, and we check the pipetting stations and heads of the machines and everything to make sure that everything goes well. But there's not big changes are expected in the next few data.
Understood.
Yes.
So question from Christoph van Schil pertaining to the data you showed on the analysis of Selenon. So they ask, what was the actual reason do you believe for why exon 1 was not covered with the automatic workflow, and what adaptations did you have to make here?
So the adaptation is in the library prep, especially after the hybridization. There you have a washing procedure using the beads, and we adapted the official protocol that was given by Twist to our own protocol, and there we saw an increase, but we lose some of the some of the targets are missed or not well represented in the pools after the enrichment. Okay. So we needed to adapt the protocol there.
Okay. And I guess a sort of slightly related question from Joe Montfort, asking how important is the fragmentation method, so enzymatic versus mechanical shearing, in getting consistent exon 1 data?
So the enzymatic shearing gives us good data. That's what we've seen when we use the manual workflow. I don't think that the mechanical shearing is actually the problem here. I think the mechanical shearing is performing well, but it's more in the steps after the shearing and the hybridization that probably needs to be tweaked to make sure that the data or the reads are represented in the exomes.
Understood. Okay. A question about the data you showed on CNVs. So how do you deal with false positive CNVs? Do you have I guess it's a big question.
Yeah, that's a big question. False positive. So that is something. So the conifer tool that we use for CNV detection is actually quite a conservative caller, and we look at the majority of the CNVs using fairly large regions. So small exonic deletions will not be picked up by the conifer tool. We need at least 3 or more targets per call. So false positive rates are not that big of an issue with CNVs. If we use other callers like exome depth, then it's probably a much bigger problem than with the conifer tool. Actually, the CNVs must be fairly large to be picked up by conifer.
Okay. Just a question from an anonymous attendee, just taking a step back, just asking how many robots are you actually using during this workflow? So in other words, what proportion of this workflow is automated versus manual human involvement with the sequencing? So obviously there's still some degree of manual involvement, but how does it break down?
So the workflow is so the library prep in that sense is more or less automated. Of course, the DNA isolation is automated, but then we have to transfer the plates to the robots that are in place for the library prep. Shearing is on deck. PCR steps are on deck. We do some QC steps. These are done manually. So checking if the library is well made, if the DNA concentration after library preparation is okay, is checked, but these are manual operations. But the rest of the library prep, of course, is more or less fully automated. Other things are, of course, that we need to pool the data set to such an extent that we can load it on the NovaSeq, and that is almost finished to prepare the pooling automatically, but that at this moment is still a manual process.
Okay, and on that note, actually, so another question regarding the enrichment: how many samples do you pool together?
So that depends a little bit on the flow cells that we use and the number of samples that we have. We have enough barcodes. That is not the issue. So on an SP flow cell, we run routinely 32 samples. We think we can increase that, but we still need to validate that and show that it's possible. It might be very well possible to run 40 samples or even 48 samples, but that needs to be checked and well validated before we can go there. On an S1 flow cell, we actually pool 64 samples. So that is routine. If the question involves pooling samples during the library preparation, that is not done yet. So our workflow is still a single plex workflow, meaning that we make one library per sample. But we hope to automate the pooling strategy for the samples to an 8-plex workflow, where we combine 8 samples after certain steps in the library preparation and finish the library preparation afterwards. But that is something that is under development, and currently we just run one library per sample.
Brilliant. Well, Marcel, we're coming up on time and there's still quite a few questions. So I think it's okay. I might suggest that we follow up some of these by email.
Sure.
So thank you to everyone who's sent us in questions. They range from the general to quite technical. So I think for some of them, you'll probably want to spend a bit of time going back to the data to answer them. Okay. So once again, thank you so much for joining us today and giving us an overview of the work in your lab and also ultimately the success that you've had with the twist exome. So I'm delighted to see that and really do wish you every success with continuing use of this workflow and also, as you say, bringing more indications into the rapid turnaround time system. So all the best. Thank you again, Marcel. And just a reminder to our attendees that when we finish, we'll be forwarding you on to a very short survey. We'd be immensely grateful for you to give us your feedback. And we'll look forward to seeing you at the next webinar. Thank you so much and have a very good day.